Before we move further, let's have a recap of what we studied on the last session. I'm sure you have prepared the topics which were taught to you very well. In the last session, we studied about respiration. We studied that in respiration, food is used up and energy is produced. I hope you remember the definition of respiration. Respiration is the biochemical process in which glucose is oxidized or broken down in the presence of oxygen to liberate energy. Now, in the last session, we also studied that whether plants breathe or not. We have studied that technically plants do not breathe, but they respire. Like animals, they use oxygen to break down sugars to release energy. This energy is used to carry out all the life processes. These include repairing and replacing tissues and building complex molecules such as proteins. During respiration, plants release carbon dioxide and water vapor like us. We also studied that respiration is of two types: external respiration and internal respiration. Internal respiration is also called as cellular respiration. We know that it takes place inside the cell. That is why it is called as cellular respiration. Cellular respiration has two kinds: anaerobic respiration and aerobic respiration. We have studied that external respiration involves exchange of gases between the organism and the external environment. The plants obtain oxygen from the environment and return carbon dioxide and water vapor into it. This mere exchange of gases is termed as external respiration. The biochemical process which occurs within the cells and tissues and oxidizes food to give energy is called as cellular respiration. Various enzymes catalyze the process, and depending upon the utilization of oxygen, the cellular respiration is of two types: anaerobic respiration and aerobic respiration. I'm sure that you remember what is aerobic respiration. Aerobic respiration is a type of respiration which leads to complete oxidation of food, or you can say that organic substance. And this takes place in the presence of oxygen. It releases carbon dioxide and water and a large amount of energy. Such type of respiration generally takes place in higher organisms. Aerobic respiration occurs in three stages: glycolysis, citric acid cycle, and electron transport. Now, aerobic respiration is carried out throughout the day and night. In the light, photosynthesis is faster than respiration, and the carbon dioxide produced is quickly used up by photosynthesis. Do you remember what happens in anaerobic respiration? Anaerobic respiration occurs in complete absence of oxygen. It occurs in many tissues of higher plants, seeds in storage, fleshy fruits and succulent plants such as cacti temporarily take to a kind of respiration. Generally occurs in lower organisms like bacteria and fungi. This results in incomplete oxidation of stored food and formation of carbon dioxide and ethyl alcohol, and sometimes also various organic acids such as malic, citric, oxalic and tartaric acids. Very little energy is released. The equation for this is C6H12O6. Without oxygen, it makes C2H5OH, that is ethyl alcohol, plus carbon dioxide plus energy. This process of oxidation in microbes is known as fermentation. It is quite similar to anaerobic respiration in case of higher plants. Now let's see how anaerobic respiration takes place in animals. In animals, glucose is anaerobically converted into lactic acid and energy is liberated. The formula for glucose is C6H12O6 and the formula for lactic acid is C3H6O3. And the amount of energy liberated is 150 kilojoule. In yeast, glucose is converted into ethanol and carbon dioxide and energy are liberated. Formula for anaerobic respiration in yeast is C6H12O6 gets converted into ethanol, that is C2H5OH, plus carbon dioxide plus 210 kilojoule energy is liberated. Now, why is aerobic respiration more efficient than anaerobic respiration? Have you ever thought of it? In aerobic respiration, glucose is oxidized to give carbon dioxide, water plus 2830 kilojoule of energy. But in anaerobic respiration, glucose gets converted into ethyl alcohol, carbon dioxide and 210 kilojoule of energy is liberated. And also when lactic acid is formed, then 150 kilojoule energy is liberated. So you can see here the difference in energy level. In aerobic respiration, more energy is liberated, whereas in anaerobic respiration, less amount of energy is liberated. So we see that in aerobic respiration, more energy is released than anaerobic respiration. Now we'll talk about fermentation. When oxygen is not available anaerobic respiration, fermentation can follow glycolysis in order to continue to produce energy. This is not as efficient as aerobic respiration and produces far fewer ATPs. Two types of fermentation take place. One is lactic acid fermentation and the other is alcoholic fermentation. Let us revise basics of respiration. Food is used as source of energy. Here, the source of energy is food. Oxygen is needed for the oxidation of food. Thirdly, energy is released as a result of oxidation of food and trapped as ATP. If you remember, ATP means adenosine triphosphate, which is also known as the energy currency of the cell. It provides immediate energy to the cells. Carbon dioxide and water are produced as end products. So, lastly the formula goes as C2H12O6 plus 6O2 with the help of certain enzymes gets converted into carbon dioxide, water, and energy is stored in ATP molecules. After refreshing all about respiration, let us perform certain experiments on respirations. This is activity 3.6 from your book, which is given on page number 44. And the aim of this activity is to show that germinating seeds respire. To perform certain activities, we need materials, and the materials needed are germinating seeds. See, you can see over here, germinating seeds, freshly prepared lime water. This freshly prepared lime water will be taken up in a test tube. This glass tube is called as a test tube. Test tube stand. This is a test tube stand. A conical flask and a connecting tube. This cone-shaped flask is called as the conical flask, and this is a connecting tube which is bent at two ends. The procedure for this activity goes like: take a test tube stand, conical flask and connecting tube. Set up the apparatuses as shown in the figure. Keep some germinating seeds in the conical flask like this. Pour some freshly prepared lime water in the test tube this way. Keep the whole apparatus this way for 1 to 2 hours. Why have we kept it for 1 to 2 hours? We are keeping it for 1 to 2 hours so that the seeds get enough time to respire. Observe what happens to the color of lime water. After 1 or 2 hours, you should observe the lime water which is kept in the test tube. Now, observation. What you will observe? You will see that the lime water turns milky. The color of lime water will be milky now. Conclusion: this shows that germinating seeds respire and produce carbon dioxide. The carbon dioxide moves through the connecting tube into the test tube containing lime water. These are the seeds. When the seeds respire, during the respiration, carbon dioxide is emitted. So, carbon dioxide gas will rise through this bent tube, move from here, and it will move through this bent tube and mix with the lime water kept here and turn the lime water to milky. Why the lime water turns milky? A question you may have in your minds. Because germinating seeds carry out their respiration process at cellular levels. Means it takes place inside the cells to be able to generate energy for growth and maturation. This we have studied, that the plants use the energy for various life processes. So, the seeds will use this energy for their growth. They do not yet have the ability to carry out photosynthesis. At this stage, the seeds are not able to respire. They are not able to photosynthesize. they are able to respire only. So as a result of respiration, carbon dioxide is released by the seeds, which turn lime water milky or white. The question arises, why germinating seeds are used for this experiment? Because germinating seeds respire a lot more than a plant that has already grown. A new plant will require more energy and oxygen to get started. Now we will be performing activity 3.7, which is given on page number 45 of your book. The aim is to show that heat is released during respiration. Now we'll see what materials are required for this experiment. We need germinating seeds. We need cotton to plug the flask like this. 2 round bottom flasks. These are called as round bottom flasks because you can see their bottom is round. And 2 thermometers. These are the thermometers. You might have seen thermometers at home. Procedure. Take some germinating seeds. Like this, we'll take some germinating seeds and boil half of them. Put the 2 types of seed into separate round bottom flask marked A and B. Like one here we have marked as A and the other one as B. A contains uncooked seed and B contains boiled seeds. Insert a thermometer in each flask and seal the mouths of the 2 flasks with cotton wool. We will insert one thermometer in flask A and the other in flask B and plug their mouths with cotton like this. Note the readings of thermometer in the 2 flasks. We will initially note the reading in both the thermometers and leave the flask for 3 to 4 hours. Now this we are doing so that the process of respiration takes place till 3 to 4 hours. Again note their temperatures. After 3 to 4 hours again we will note the rise of mercury in these thermometers. Observation. The temperature of flask B containing the cooked seeds remain the same. Now in this flask, the thermometer that was kept showed the same reading as it was showing earlier. Whereas the temperature of flask A containing the germinating seeds rises. Now the temperature of this flask shows a rise in flask A. Conclusion. This shows that heat is produced during respiration. If you remember the formula that I told you before, that during respiration nearly 2830 kilojoule energy is liberated. This energy is the heat energy. So due to this heat, the temperature rises in flask. See, this slide displays why the temperature rises in flask A. As I have just now told you that the heat is the energy which is released during the process of respiration. The germinating seeds respire, in which glucose is broken down and the energy is released, which is liberated in the form of heat. This is what I told you, that it gives out 2830 kilojoule energy. But then a question arises, that why the temperature did not rise in flask B? That means that in flask B we had kept cooked or boiled seeds, and you know that after boiling the seeds get killed, and so they did not show any sign of respiration. They did not respire, they did not give out any gas. This is the reason that the temperature did not rise in flask B. Now we will study another activity which is given in your book, and that is activity 3.8, given on page 45 of your book. Now the aim of this activity is to show that plants take in oxygen during respiration. Materials required are germinating seeds, KOH pellets. KOH means potassium hydroxide, mercury, and 2 beakers and 2 respirometers. This is the respirometer. Here inside it we'll be keeping KOH pellet or KOH crystals, and in a flask we'll take some mercury. This is the stand which is used to hold the respirometer. Inside this respirometer we will keep germinating seeds. Procedure, or how we'll do it, method of doing. Take some germinating seeds in the bulb of respirometer. This swollen area, bulb-like area, is called as the bulb of respirometer, in which we'll keep the germinating seeds. Insert the tube of respirometer into a beaker containing mercury. This is the tube of the respirometer which will be inserted in the mercury. Introduce a pellet of KOH in the setup. A small crystal of potassium hydroxide is kept in this tube. Set up another experiment without any seed to act as a control, and we'll set up another experiment, another setup in which we will not keep the germinating seeds in the bulb of the respirometer, just to see that what happens. Observe after 1 day. We'll leave it for 1 day and observe both the setups after the day. Observation: the level of mercury rises in the respiratory tube of the setup that has seeds, but remains the same in control setup. Control setup means in which we did not keep germinating seeds. What do we conclude? This is because the oxygen inside apparatus is used by the seeds during respiration. As a result, air pressure inside the bulb is reduced, raising the level of mercury. Now see, here we had kept germinating seeds, and you know that seeds respire. During the process of respiration, they take in oxygen and they give out carbon dioxide. So when the seeds were respiring, they used up all the oxygen that was present in the bulb or in the tube of the respirometer, and this created a pressure, this created a vacuum, and thus caused the mercury to rise up in the tube of respirometer. So we have revised the process of respiration, and we have also studied that the external and internal factors that are necessary for respiration, how they affect a plant. Now we will see that how certain factors affect the process of photosynthesis. So before studying that, first I would like you to recall whatever you have studied about photosynthesis. So let us recall it. Photosynthesis is the process by which green plants, also algae and some bacteria, make their food from carbon dioxide and water with the help of sunlight and chlorophyll. So that is what is photosynthesis. Now the process of photosynthesis is a chemical reaction. It is the most important chemical reaction in the world. Why is it so important? Because had there been no photosynthesis, there would have been no life on earth. Because after photosynthesis, the plants produce food for themselves as well as they produce food for other organisms. And by gaining that food, the heterotrophs get energy and are able to utilize that energy for their life processes. Now we'll talk more about photosynthesis, that why photosynthesis is a photochemical process. As I said earlier, photosynthesis is a photochemical process because light is required for some of the reactions to occur. The word photo means light. So that part of word refers to the fact that the light dependent phase is initiated by the absorption of light by chlorophyll. Now let us note that although the word photon is a measure of light, that is not the word incorporated into photosynthesis. Also remember, the synthesis means to make or to manufacture. So that part refers to manufacturing reactions of the light independent phase from which organic molecules are synthesized or made. And what is that organic molecule? That is glucose, C6H12O6. Now, are you able to tell me what are the raw materials needed for photosynthesis? Yes, the raw materials needed for photosynthesis are sunlight, water, carbon dioxide and chlorophyll. A commonly used but slightly simplified equation of photosynthesis is 6 molecules of carbon dioxide plus 12 molecules of water plus photons. Photons means light energy is equal to C6H12O6, that is glucose, 1 molecule of glucose plus 6 molecules of oxygen. Now, where does photosynthesis occur? We all know that photosynthesis occurs inside the leaf. But where inside the leaf? In chloroplast, in the cells of leaf. In most plants, photosynthesis occurs primarily in the leaves in the chloroplast. And what is chloroplast? What is the structure of a chloroplast? A chloroplast contains stroma, which is a fluid, and grana, which is made up of stacks of thylakoids. The thylakoids contain chlorophyll. Chlorophyll is the green pigment that captures light for photosynthesis. Now you can see this is the structure of a chloroplast. This is how a chloroplast looks. And this is a diagrammatic figure of a chloroplast. The chloroplast is bounded by two membranes, the outer and the inner. Here you can see this is the outer membrane and this is the inner membrane. And see here, this is the fluid part which is shown here with fluorescent green color. This shows the stroma part. And here the stroma is a fluid part, liquid part. And this coin-like structure is a thylakoids. So many thylakoids are stacked one on top of the other, forming a grana. Now we have seen that photosynthesis is affected by two factors. One is external factor and the other one is internal factors. These factors include carbon dioxide, water, chlorophyll, temperature, etc. Now we'll study how these affect the process of photosynthesis. So we'll study them one by one with the help of experiments. Now, is carbon dioxide necessary for photosynthesis? We all know that photosynthesis is carried out in the leaf. Or we can say that the leaf is said to be the kitchen or the food factory of a plant. Now, how does carbon dioxide get inside the leaf? The carbon dioxide enters into the leaf through the stomata, which are present on the lower surface of the leaf. Let us study an activity, that is activity 3.1 from your book, given on page number 35. The aim of this activity is to show that carbon dioxide is necessary for photosynthesis. Materials required for this experiment is 2 potted plants, 2 bell jars. Why do we call them as bell jars? They are made up of glass and they are shaped like a bell. That is why called as bell jar. Other materials needed are Petri dish. This is a Petri dish, and KOH solution or potassium hydroxide solution. We also need iodine solution. We need a dropper to drop down these liquids. Now we move on to the procedure. Take 2 small potted plants and keep them in dark for 48 hours to destarch the leaves. Destarch the leaves means we know that plants prepare their food, and this photosynthesis takes place inside the leaves, and when glucose is formed, this excess amount of glucose is converted into starch and it is sent for storage to different parts of the plant. The plant uses the starch for various life processes. So when we have kept it for 48 hours in the dark, that means it will not be able to photosynthesize for 48 hours, but it will convert the starch into glucose, and that glucose will be used up by the plant. This process is called as destarching. Next, place these plants separately under 2 bell jars labeled as A and B. Now we'll put these plants inside the bell jars. As we have taken 2 bell jars, we will mark them as A and B. Place a petri dish filled with KOH crystals under the bell jar A. Now in the first bell jar, inside we will keep a petri dish in which we have kept KOH crystals, that is potassium hydroxide crystals. Place the 2 setups in sunlight for 3 to 4 hours. Our purpose for keeping them in sunlight is to enable them to prepare their food or for the photosynthesis to take place. Test the leaves from both the plants for starch using iodine solution. Now we will pluck the leaves from the plants and then we will perform the starch test as I have told you earlier. The leaf from potted plant kept under the bell jar A gives negative result for starch test. It will give a negative test because KOH absorbs carbon dioxide, and when there won't be any carbon dioxide inside the bell jar, the plant will not be able to photosynthesize. So the leaf will not give the starch test. The leaf from potted plant kept under the bell jar B gives positive result because KOH was not kept inside that and the carbon dioxide gas was available to the plant and it was able to photosynthesize. Conclusion. What do we conclude by this? The KOH or potassium hydroxide crystals absorb all the carbon dioxide inside the bell jar A and therefore for photosynthesis does not take place. This shows that carbon dioxide is necessary for photosynthesis. We'll perform a multiple choice question. Carbon dioxide enters the leaf through the dash. Now, are you able to answer it? That carbon dioxide enters the leaf through what? Through cuticle or through chloroplast or through mesophyll cells or through stomata. What should be the correct answer? The correct answer is D. Carbon dioxide enters the leaf through the stomata. Stomata. What are stomata? Stomata are the tiny pores or openings on the leaves through which gases such as carbon dioxide and oxygen can diffuse in and out of the leaf. A. If the answer would have been A, what is it? The cuticle is the waxy layer covering leaf epidermal surface, and we know that it does not take part in photosynthesis. If the answer would have been B, that is chloroplast are the sites of photosynthesis reactions. They do not allow the intake of carbon dioxide or oxygen. Now, we'll see KOH crystals are used to absorb dash. What is potassium hydroxide used to absorb? Does it absorb oxygen? Does it absorb carbon dioxide? Or it absorbs water vapor? Or the answer is none of the above. What is the answer? Can you tell the correct answer? Yes, you are right. That KOH absorbs carbon dioxide gas. Now let us find out whether light is necessary for photosynthesis or not. For this, we shall perform an activity that is given in your book on page number 39. This is activity 3.3, and the aim is to show that light is necessary for photosynthesis. What are the materials required for this activity? They are a potted plant. This is a potted plant. A black paper. This is a black paper which we will be clipping onto the leaf. Ethyl alcohol, iodine solution, test tube, beaker, petri dish, and apparatus for water bath that we have used for testing starch in a leaf. That is Bunsen burner, tripod stand, and wire mesh. How will we perform this? Take a potted plant with broad leaves and place it in a dark room for 2 days. Again, 2 days means 48 hours. As no photosynthesis takes place in the dark, the leaves will become starch free. This is also known as destarching the plant. Cover a part of any one leaf on both sides with black paper. Now we can choose any leaf and cover both the sides of the leaf with a black paper or a carbon paper. Place this plant in the sunlight for 5 to 6 hours so that the plant starts making its food. Pluck the covered leaf and remove the black paper. After 5 to 6 hours, we will pluck the same leaf and remove the black paper from it. Put the leaf in a test tube containing ethyl alcohol. We'll keep it in the ethyl alcohol solution and place the test tube in a beaker containing water and heat the water. That is, we will give it a water bath. We will never boil alcohol directly as it may catch fire. We all know, you might have studied in chemistry, that alcohol has a volatile property. That means it catches fire easily. As the alcohol begins to boil, the chlorophyll dissolves in it and the leaf becomes chlorophyll free. Take out the leaf and put it in hot water to make it soft. Keep the leaf in a petri dish and put a few drops of iodine solution over it. Now we will observe. You will notice that the uncovered part of leaf turns blue-black. This is the uncovered part of the leaf which has turned dark in color or blue-black in color, whereas the covered part turns brownish yellow. The covered part, this part which was covered with black paper, turns brownish yellow. What conclusion do we take out from it? This happens because the uncovered part of the leaf received sunlight and was able to photosynthesize and manufacture starch, which is one of the end products. Starch turns blue-black when tested with iodine. However, the covered part did not photosynthesize as it was not exposed to sunlight. That means the covered part of the leaf did not receive the sunlight and hence it was not able to make its food. Hence, it does not turn blue-black when tested with iodine. This shows that light is necessary for photosynthesis. This we come to the end of this lesson. We have completed lesson 1, that is tissues, in which we have studied about the plant tissue and different types of animal tissues in detail. We have studied lesson 3, that is plant life, in which we have studied about the process of photosynthesis and the process of respiration in detail. So we have completed the portion or the syllabus of the first semester exams. It is my earnest request to prepare well for your exams, as you have sufficient time to study. So make good use of this lockdown period. Your class notes will be uploaded on the school's website. You have to note them down in your interleaved copy in legible handwriting. You shall draw diagrams as per the need of the question. So now I conclude my session, hoping to meet you when we tide over this unfavorable period. So stay happy, stay healthy. God bless you.